finals on Friday. I guess I told you about the final exam last time. So if you have questions, what's on it, talk to me after the lecture. I'll go over that with you. I believe I told you to get this 100-question Scantron form from the bookstore. Somebody went there and said the bookstore no longer carries these. Has anybody tried to find these in the bookstore? Okay, well, if they have these, get them. But if it's true that the bookstore is no longer carrying them, then look for the green version of this. There should be either a brown or a green version in there. So I guess either one will do for the exam. But do bring a Scantron. Or I'll send you back to the bookstore on Friday at 11:30 to go get one. What else? As usual, bring your DNA technology handout to the laboratory today. I'm going to go over part of that as a pre-lab lecture. And as usual, we'll have the laboratory weekly, the 8-point laboratory weekly quiz today. Boy, what did we do last week? We did, was it electrophoresis and DNA restriction digestion? I think those are last week's labs. So whatever labs we did last week, that's what the weekly quiz will be on. Okay, well let's jump back into the genomes chapter. We started going over this, what they found when they sequenced the human genome. And one surprise was that we had a lot fewer genes than they thought that we had. But another surprise was how much of the genome was not genes. Remember that only 25% of the genome is actually DNA encoding genes. The green part of this, or genes that encode our proteins, the green part of this diagram. And the rest of it is a bunch of other types of things. As you see here, this fraction here is non-functioning genes, essentially mutated genes that aren't being used anymore. This fraction, which I think was around 5% if I remember correctly, where just large segments of the chromosomes are found duplicated throughout the genome. This tandem repeats were short segments, very, very short sequences that were repeated sometimes thousands of times in a row. Like this particular sequence, GTTAC, is found repeated over and over and over again in a row. And they mostly find those tandem repeat sequences at the tips of the chromosomes, or the tips of the chromatids. And those regions are called telomeres. And they also find these tandem repeat DNAs around the centromere region. And so what they think it is that there are certain proteins that help the chromosome function that home in on these sequences for binding to the DNA. Like I mentioned, around the centromeres, there's these things called kinetochores where the spindle fiber is attached to. The kinetochore proteins probably look for these sequences for where to attach. And at the telomeres, at the tips of the chromosomes, the DNA polymerase and primase enzymes have trouble duplicating those regions, and so you need some special enzymes to duplicate the tips of the chromosomes. And so again, those special proteins probably look for these tandem repeat sequences. And the last thing we came up to was this, that the largest type of DNA, I guess I should say, or sequences in the DNA that you find in the genome are these things called transposable elements. And they're sometimes referred to as the jumping genes because that's essentially what they do. They are segments of DNA within our genomes that can jump around. They can move from one chromosome to another chromosome, or even move to different locations within the same chromosome. And remember, in some cases, it's more like the command copy function on your keyboard, where the original piece of DNA stays stationary in its original location and a copy gets moved to a new location. And in other cases, it's more like the command X function on your keyboard, where you actually cut out, the DNA sequence actually gets cut out and travels to a new location. So these were discovered in the 1940s by this woman, Barbara McClintock, when she was studying corn. But no one really believed her at first, but she was proved to be exactly correct, not just for corn, but all species on Earth have these transposable elements. They're sometimes called lines, long interspersed nuclear elements. And there are two varieties of these transposable elements, transposons and retrotransposons. And the distinction really is what exactly moves between the chromosomes, what exactly jumps. If it's a piece of DNA that moves, that the intermediate is, and the moving form is, then it's called a transposon. If an RNA is involved in the movement, then it's called a retrotransposon. And I think I have a diagram of that. Here's a transposon. It's essentially a short piece of DNA, like you see there in the dark blue, remember? And I definitely need to get a new laser pen. And what it contains is it contains genes for enzymes. And these enzymes, when they're made into, I should say that when these genes are expressed, and you get these proteins from this transposon come back into the nucleus, look for the gene that made them. These proteins somehow hone in on their own sequence in the genome, and then they find that sequence, and in some cases they copy it, and in some cases they physically cut it out. But in any event, what you end up with is an actual piece of DNA that's floating around inside the nucleus of your cells, and then it drifts around randomly, and eventually will insert itself into a new location in the genome. The retrotransposons were even stranger still. These retrotransposons encode the reverse transcriptase protein, and so there's the mRNA that first gets made from that reverse transcriptase gene within the retrotransposon, and so that goes out of the nucleus into the cytoplasm. A ribosome translates that messenger RNA into reverse transcriptase. But it's not just any old reverse transcriptase. This reverse transcriptase has a binding site for its own mRNA sequence, the mRNA that created it. And so it finds its own mRNA, binds to those sequences, and then does what reverse transcriptase does, which is make a complementary DNA to the mRNA. So now you end up with a complementary DNA, and then that piece of DNA then inserts itself somewhere else in the genome. And I think I mentioned this, that you find reverse transcriptase genes in certain types of RNA viruses, and so they think that these retrotransposons probably originated as a viral gene that somehow became disconnected from the rest of the viral chromosome, and so in a sense it's now almost like a mini virus that's trapped inside the gene, trapped inside the nucleus. It can reproduce itself inside the nucleus and move around, but it can't escape from the nucleus because it no longer has capsid proteins or anything like that it would need for it to move from cell to cell. And so don't get me wrong. I'm not saying these are viruses, but they're believed to have originated as a viral gene. Let's see. There are also these things that are called signs, which are short interspersed nuclear elements. And a sign is essentially a fragment of a retrotransposon. Imagine a retrotransposon that was shortened or truncated so it did not encode the full reverse transcriptase. It does not make any functional reverse transcriptase. Like this. And so it can never make any reverse transcriptase protein, but as long as there's a retrotransposon anywhere else in the genome that is making reverse transcriptase, that same reverse transcriptase enzyme will come back into the nucleus and it will find the mRNA from the sign, and since it recognizes those sequences, since those are at least part of a reverse transcriptase messenger RNA, it will find those and then do what reverse transcriptase do, which is make a cDNA copy of it, and so this will then insert itself into the genome. And so if you think about it, in a way, this partial retrotransposon is going to be more successful than the actual retrotransposon just because it's shorter. The reverse transcriptase can make DNA copies of it much more quicker just because it's not as long. And so it essentially outcompetes the real retrotransposons, the full-length retrotransposons. And there's actually many different of these signs that they find in our genomes, but this ALU one is one of the most abundant of the signs. And not that you have to know the sequence, but bracketed between the red sequences there is the ALU sequence. I don't know where they came up with that name from, but it's just one of the signs. And so it is the first part of a reverse transcriptase gene, but not the whole thing. And you find these ALU sequences all throughout our genomes, over 500,000 copies total. So just this sequence alone makes up about 10% of the human genome. And I think maybe Juan asked me last time, like, how often do these transposable elements jump around in our chromosomes? And I had trouble finding any sort of exact number for it. All the textbooks and the websites mention these transposable elements, but nobody says, well, how often do they jump? But one reference did say about 1 in every 200 people has a new ALU insertion, a new sign insertion somewhere in their genome. And so that means to me if only every 1 in 200 people has one of these, then they don't jump around all that often. For the most part, they're stable, but they do occasionally jump from chromosome to chromosome. Okay, well, let's see. Maybe a good thing to mention here at this point is do these transposable elements, well, maybe before I even answer that, I was going to say, do they do us any good or do they do us any harm? Maybe one thing I should mention is how do we compare to other species? So in our own genomes have around 44% transposable elements. Most of them are made up of transposons and retrotransposons, like you see in this part of the 44%. But about, I don't know, a little over 10% of our genome are these signs, the shortened ones, ALU and other signs. But anyway, if you take it all told, it's around 44% of our genome, which is more, almost double the amount of our genome that's genes. So it's almost like the word for our genome has the word gene in it, but that's almost like a misrepresentation because it's not mostly genes. More than anything else, it's these transposable elements. We have more of these than other species. And let me write down here some the amount of genomes that other species have of their transposable elements. And you don't have to memorize these, but in bacteria, only about 1% of their genome are these transposable elements. So we have 44 times as many as bacteria. For flies, it's around 3%. For nematodes, which is a type of worm, it's around 7%. And for mustard plants, it's around 11%. So as I said, you don't have to memorize any of those, but the point here is we have a lot more transposable elements in our genomes than a lot of other species do. So does having all this jumping genes do us any good, or is it harmful to us? Well, the answer is a little bit complex. To begin with, I'll address the harmful part. Potentially they are harmful because when these transposable elements jump around in our genome, the site where they splice themselves into is essentially random. So they can land anywhere in your genome, including one of your functional genes. And if you have a nice functional gene and suddenly this transposable element whammo jumps in the middle of that gene, it's probably going to disrupt that gene. Here's an illustration of that. There's a I guess I'm using a retrotransposon here, but it could be a regular transposon also. So it gets transcribed into a piece of DNA like this, and then that kind of randomly drifts around in the nucleus and eventually inserts itself into a gene right there. And as it does, it's going to open up the DNA there and insert that new copy, and that's probably going to wreck the gene in the sense that you're disrupting the codon flow of that particular gene. So certainly most times it'll just, when this happens, if the transposon lands in a gene, it's going to make that gene non-functional. If you make random changes like in your computer, open it up and just grab wires and move them around randomly, you're probably going to make your computer just stop functioning. It might end up altering the function of the gene. That is, if it just ends up altering a few amino acids encoded by the gene, you might get a protein that has some new function that it didn't have before, or it might potentially increase the function, make it a better enzyme, but that would be a very rare instance. That would be like, again, monkeying around randomly with your computer and somehow making your computer work better. Not likely, but it could happen. Also, it could leave the protein encoding part of the gene alone, but if it lands in one of the areas that affects the expression of the gene,